大家好，今天我们生物医药主题第二位要出场的是关泰林博士啊，等一下，大家好，今天我们创新创业讲座，第二位出场的是关泰林博士。那关泰林博士呢，也是剑桥大学国际奖学金的获得者，研究项目为化学选择性和位置专一性蛋白修饰，并与英国医学研究委员会分会的这个生物学实验室和剑桥大学生物化学系的研究小组保持着密切的合作。那下面让我们掌声有请我们的关博士为我们带来精彩的演讲。
Thank you very much, and thank you to Michael for that excellent overview of the pharmaceutical industry and how the use of flow chemistry can actually help speed up the process of drug discovery and manufacturing. So today my talk will actually focus on the other aspect of the pharmaceutical industry, which is the biopharmaceuticals. And my talk will give a brief overview of the different functional classes of protein therapeutics, as well as trying to give you a flavor of how chemical synthesis and chemical strategies can help improve existing biopharmaceuticals and potentially generate some new drugs. So first off is, what is biopharmaceutical? So according to a commentary in Nature Biotechnology, a biopharmaceutical is a pharmaceutical that is inherently biological in nature and is manufactured using biotechnology. So these would include, say, peptides, recombinant proteins, antibodies, antibody-drug conjugates, etc. It is important not to confuse this with the term biosimilar, which is actually a copy of an innovator product that is manufactured by a different company, so a generic for pharmaceutical drugs. A biopharmaceutical has a huge impact on the whole industry, commanding roughly over $160 billion per year. And that is roughly 20% of the market. And it has an annual growth rate of 8%, which is the fastest growing part of the industry. One of the key differences of a biopharmaceutical is that it is structurally very complex, giving it very high specificity and potency with minimal side effects. So, this diagram summarizes the differences quite nicely. If you consider the complexity between a car and a bike, so a car and a bike, so roughly 3,000 pounds to 20 pounds. That is the similar differences to a peptide drug, somatropine, and aspirin. So somatropine is a growth hormone, 191 amino acid long peptide drug, and aspirin, as you all know, is a very well-known painkiller, consists only of 21 atoms. However, if you actually increase the complexity to, say, Herceptin, so Herceptin is an antibody used to treat breast cancer, and it consists of 25,000 atoms, and that is roughly, say, a business jet complexity difference compared to a bike. So we're talking a completely different set of pharmaceuticals here, and it's best if you keep that in mind. So what are the advantages of having such a complex structure? So at this point, I would like to stress that these complexity is not just plucked out from thin air. These are scientifically hypothesized, designed, and tested. So these complexity actually give us extremely good efficacy with minimal side effects as compared to small molecule drugs. And when you compare the two advantages together, you would generally get a faster FDA approval as well. In addition, from a financial perspective, due to the protein's unique form and function, you can actually get very good far-reaching patent protection. That is probably not very easily done with small molecules. So biopharmaceuticals normally use naturally occurring products, proteins, as therapeutics. and since the completion of the Human Genome Project back in 2003. It has opened up a huge potential in personalized medicine, such as gene and cell therapy. Lastly, there are over 40 different genes coding for functionally unique proteins, and this represents a large pool of untapped possibilities for new and exciting biopharmaceuticals. So, first, let me describe to you the different functional classifications of protein therapeutics. In the first class, we have what we describe as the classic paradigm in which a specific endogenous protein is deficient, and the deficit can be remedied by an exogenous protein. That sounds a bit complicated, so let me give you an example. So we have humulin, developed by Lilly, and it treats diabetes. In diabetic patients, they lack a protein called insulin, which is responsible for regulating glucose metabolic pathways. So if a patient does not have insulin protein, what they would do is they would inject themselves with humulin, which is a recombinant insulin, in order to alleviate diabetic symptoms. Other classes of protein therapeutics in this functional class include interferons and growth hormones. In the second class, we have protein therapeutics with special targeting activities. So what we're talking about here is antibodies. Antibodies can target very specifically to your diseased cell, and then can switch on the body's immune system to neutralize the foreign virus or pathogens. In addition, you can also conjugate your antibody to a therapeutic drug, giving you antibody-drug conjugates. And these can allow the cytotoxic drug to target your pathogens in a targeted manner. I'll talk about this in a bit more detail later on. And in the last class, we have vaccinations. Traditionally, vaccinations against various diseases are achieved by injecting heat-killed or attenuated forms of pathogens into the body. Unfortunately, there will always be some sort of unavoidable risk involved with this. However, in protein vaccines, what we can do is we can actually take the antigen part of your virus and then make it into a vaccine. So the virus will never enter your body, only the antigen would be injected into your body in order to train your body's immune system against a potential viral attack. Moving into something a bit more towards personalized medicine, we have prevention here, where we synthetically train the human body to target its own cancer cells. So it consists of 3 basic steps. First, the patient's blood would be subjected to leukapheresis, where they extract the white blood cell. And then the white blood cell can be infused with the recombinant protein PAP-GM-CSF. Is an antigen against prostate cancer. And then once the white blood cell has been infused with this external recombinant protein, it can then be injected back into the patient, which then has its immune system trained against the cancer cell. So, given the success of biopharmaceuticals, what are the challenges in the industry? So usually, biopharmaceuticals suffer from very poor solubility and stability. The distribution and administration of the drugs are also very difficult. In addition, we need post-translational modifications in order for the therapeutic to be physiologically active. So if you need to glycosylate or phosphorylate site-specifically, how do you do that is a challenge. In addition, we have heterogeneous mixtures of biopharmaceuticals. This is a major issue in the industry because it is very difficult to optimize and characterize these mixtures, and you often result in low FDA approval. It's something similar to traditional Chinese medication, where there are a number of chemicals that are working in tandem to elicit therapeutic effects. So how do you determine which one of these chemicals or which one of the protein therapeutics is actually responsible for the effect? That's the challenge there. So if you look at this diagram, I think this sums up the problem of heterogeneity quite nicely. If you look at an antibody, we have 40 lysine residues. And if you use classical lysine conjugations, you can get a mixture of drug-antibody ratio from 0 to 6 with a lot of variations up top. That is very difficult to characterize and analyze. However, if we can actually site-specifically modify the antibody at those two sites only, then the picture becomes a lot clearer, and it's much more easy to analyze and optimize these biopharmaceuticals. So how do you do this? So my PhD project mainly focuses on this area, which is bioorthogonal chemistry. This term was first coined by Professor Caroline Bertozzi at UC Berkeley back in early 2000. And in its core, it is chemical reactions that do not interfere with native biochemical processes. So if you imagine a chemical reaction inside the body, we have a unique functional group labeled in blue here. And if you interrogate this functional group with a complementary functional group, yellow, the two can react in physiological conditions to give you your stable covalent linkage there, joining your biomolecule to the drug or fluorophore in question. For a chemical reaction to be classified as a bioorthogonal reaction, it must have fast kinetics, biocompatible, and the functional groups must be very selective for each other. In addition, these functional groups must be easily accessed by protein engineering. So after decades of research, there are now a wide range of different chemistry that can be classified as bioorthogonal reactions. And they can be generally summarized as 3 groups. So we have the condensation reactions, such as the ketone and aldehyde, condensation reactions with amino oxy or hydrogen functional groups. We have the cycloadditions, so we have the famous copper A-side alkyne click reactions, as well as the Diels-Alder cycloadditions between a strained alkene or alkyne and tetracenes. And lastly, we have the metal-catalyzed reactions, such as the cross-metathesis reactions using ruthenium catalyst or the palladium cross-coupling reactions. So despite a huge number of reactions being developed, there are still further needs for more bioorthogonal reactions because there is no one conquering reaction that can be applicable for all biological systems. So, in light of this, one of my PhD projects is the development of a new bioorthogonal reaction, and it's the hydrosilation, which utilizes unreactive alkyne group and a hydrosilane group as coupling partners. So the rationale is that silicon and carbon have very similar chemical properties because they're in the same group in the periodic table. So it is less likely for the protein to reject this modification or for the silicon to alter the protein's function. However, one key difference is that the silicon-hydrogen bond is very weak, making it very reactive to the ruthenium catalyst, and therefore very low catalytic loading is required to facilitate the hydrosilation reaction. Furthermore, what makes this reaction very significant is that it's got orthogonal reactivity towards other bioorthogonal reactions, which means there is potential for simultaneous site-selective conjugation of different chemical groups onto a single therapeutic biomolecule. So, to highlight the different strategies for site-selective protein modification, I'm going to use the antibody-drug conjugate as an example platform. So listed out here are 5 of the currently top-selling antibody therapeutics. So we have Humira, Remicade, the famous Herceptin for breast cancers, and they all generate multi-billion-dollar revenues per year.
Avastin is for treating macular degeneration.
Avastin has got multiple uses, and one of them is angiogenesis inhibitor as well for cancers.
Well, yeah.
Yeah.
That's for inhibiting blood vessel growth.
Yeah, blood vessel growth, exactly. So, antibody is a proven therapeutic in the biopharmaceutical industry already. So, how can we enhance its therapeutic properties? And that is by conjugating drugs onto them. So there's a number of factors to consider when you're making an antibody-drug conjugate. What sort of antibody should we use? What's the target? What sort of drugs are we going to conjugate onto the antibody? What sort of linker are we going to use? Is it going to degrade in the lysosome? Is it going to degrade before it gets to the target cell? And what sort of tumor, what sort of disease we're going to target? These are all questions we have to ask. So, if you remember from this previous diagram, we do not want to do that. That's going to give you a mixture of antibody-drug conjugates, and it's going to be very difficult to isolate, to administer as a heterogeneous mixture is never a good idea. So what we ideally want is to somehow pick out a certain site in the antibody and then conjugate your drug onto it, site selectively. Very important. Yeah, so site-specific conjugation would minimize heterogeneity, more predictable pharmacokinetic properties, and is much higher consistency when you manufacture these drugs on a batch-to-batch. It will also facilitate optimization, achieving similar efficacy at a lower drug dosage, which means there's less chance for the patient to suffer from cytotoxic effects by having too many cytotoxic drugs in your body. So, a classical method for these sort of conjugation is to use cysteine chemistry. Cysteine it normally exists as a disulfide bond, and what you can do is treat your whole antibody with DTT, which reduces your antibody to give free sulfur. And DTT would reduce the disulfide linkages, and then when you react it with your maleimide, it would do a 1,4 microaddition reactions to put 8 of your cytotoxic drugs onto your antibody. 8. Still not perfect. We want to put 2 or 1. So what we can do is use a new version of cysteine chemistry called Thiomab. So what scientists at Genentech did was that they site-specifically mutated your antibody to incorporate a cysteine molecule, say, at that position, solvent exposed. What it would do is reduce all the cysteine using DTT and then reoxidize the antibody specifically using dihydroascorbic acid or copper sulfate. That means all the interchain sulfur bonds would be formed again, leaving your solvent-exposed cysteine for reaction. So therefore, when you treat your antibody with maleimide, only those two would react, giving you high purity of drug-to-antibody ratio 2 antibody-drug conjugates. Another way to do this is to incorporate a natural amino acid into your antibody. So this method is a bit more challenging because you need more protein engineering techniques. But if you can do it, you can put, say, a ketone group, and then you do a condensation reaction with your drug. You can form a stable oxime linkage there, and you can do this site-specifically as well. And you can also incorporate a selenium into your antibody, and again, because selenium has a very unique nucleophilic property, it would only undergo 1,4 addition with your maleimide, giving you site-specific conjugations there. So these are some of the strategies for that. And the last method I'm going to talk to you about today is enzymatic conjugation. So human antibody immunoglobulin G has a conserved glycosylation site. So if you actually treat your antibody with modified sialic acid, which contains an azide, it can react with your strain cycloalkyne drug to conjugate your drug onto the glycosylation sites of the antibody, site-specific. Other ways that you can do it is using transglutaminase, which recognize this peptide tag, LLQGA, and it would do a transaminate basically, with small molecule containing a primary amine group. So again, you can put them on site selectively, and similarly, if you use a formal glycine generating enzyme, which recognizes this peptide sequence, it can put an aldehyde here, which would then react in condensation manner with this group to put a drug on site specifically. So let's use a real-life example to talk about the significance of site specificity conjugation. So we have Kadcyla. Kadcyla is one of the 3 marketed antibody drug conjugate currently currently FDA approved. And it uses the Herceptin antibody here, which recognizes the HER2 metastatic breast cancer cells. And it's been developed by Genentech and Lonza. What's different between Kadcyla and Herceptin is that we have a linker and a DM1, which is a cytotoxic agent targeting microtubule formation. And it's been only approved back in February 2013, and since then, within this period, it already generated over $160 million in sales, with a projected $2.5 billion per year. So how does Kadcyla work? Kadcyla works by binding to HER2 antigens on the breast cancer cell, and once found, the antibody can elicit immune response from the body, which can neutralize the cancer cell. In addition to this standard antibody mechanism, the antibody can also be internalized, and once it's in the lysosome, DM1 can be released and stop microtubule formations within the cancer cell. So basically, we're destroying the cancer cell from within, both from the outside and from within. So it's a double mechanism working in parallel. So currently, Kadcyla is generated by using lysine chemistry, which means there's a heterogeneity mixture of antibody drug conjugates. However, a study done at Cellarin Therapeutics shown that if different cells conjugated at different sites of the antibody would have different effects. So they used the thiomap technology, and they chose 3 different sites on the antibody with different solvent accessibility. So we have low, medium, and high. And what they found out is if the drug is conjugated at a very high solvent accessible site, it can actually undergo Malay exchange, which means the drug is released before it even gets to the cancer cell. So we have protein conjugate stability decrease and a decrease in therapeutic activity. However, if you conjugate at a site that is positively charged environment, it actually undergoes the succinimide ring hydrolysis, therefore increasing their protein stability and increasing therapeutic activity. So this study actually highlights the importance of where you put the drug. You don't want to put it everywhere. You actually want to put it at a place where you know it's going to be therapeutically favorable. So in conclusion, site-specific conjugation is a very important strategy for improving and modifying existing therapeutics. You can put chemical groups such as polyethylene glycol groups site-specifically onto the protein, which would increase its solubility and decreases immunogenicity, which is not good. In addition, you can also put post-translational modifications site-specifically onto your biopharmaceutical, which can bypass the need for specific cell types that can incorporate these PTMs in the manufacturing processes. And lastly, we can site-specifically conjugate chemotherapeutic chemical drugs to clinical proven antibodies, like I've talked about in the antibody drug conjugates. This would definitely increase its efficacy and selectivity. Somewhat further down the line is the use of these technologies to do novel biopharmaceuticals, which can allow us to tap into previously unexplored drug space. So we can generate some sort of chimeric therapeutics, part biologics, parts chemical, so they would definitely have some very interesting properties, as well as fusion biomolecules, something which is not naturally occurring, such as protein DNA conjugates with weird linking molecules, or protein-protein fusion, not at the N or C terminus, etc. So I think they would definitely have a huge impact in the industry when the science and research matures in the near future. So, medicine is approaching in a new era where strategies to manage diseases is coming from the protein level and genetic information. The high efficacy and safety, as well as its ability to target many of the orphan diseases, allow it to command very high prices where the top 15 biopharma products enjoy an annual revenue of over $2 billion. However, there are a lot of challenges in the field. It is very difficult to reliably reproduce biological macromolecules because of its complexity. In addition, if you want to start making your biomolecules, you need to invest a lot in costly facilities. Raw materials are very expensive, and it's a very long process. The manufacturing process has a long durations, and it's often very low yielding as well. Lastly, we have the development of the biosimilar industry. So there's an upcoming patent cliff, putting around 36% of the market at risk. So there are over 700 biosimilars in the development pipeline right now. But thankfully, the industry realizes these challenges, and it's actually one of the most R&D intensive, with some of the companies spending over 40% of its R&D budget in biopharmaceuticals. From a China perspective, the biopharmaceutical industry is actually one of the 7 key areas for development in the 12th five-year development plan, with numerous financially favorable policies. There are already a number of biotech companies conducting innovative research in China, such as New Biotherapeutics, Silva Biotherapeutic, and Sinovac Biotech. The key driving force for innovation within the sector is streamlining the manufacturing process, allowing it, improving in analytical methods, and ultimately reducing cost. The ability to chemically modify biomolecules will undoubtedly simplify both the manufacturing processes, and if you can do it site-specifically, simplify the analytical method as well. I wholeheartedly believe that synthetic chemistry, and in particular bioorthogonal chemistry, will have a huge impact in the field. Thank you very much.
Now it's the time for question.
Just a quick comment. Back one slide.
Back one slide.
When you talk about solubility,
Right.
you really need to talk about solubility against either water or lipid, because the two are different things.
Yeah, the two are very different
Yes,
things.
and also solubility in lipid is actually quite an important design goal for drugs, because anything that's soluble in lipid, that means it's get collected in the liver,
Yes.
where the fatty cells are.
Yeah.
And if it's toxic, then that means it will
It's
affect
not
the
very
liver.
good for you.
Yeah. So when you're talking about solubility, you
We need to expand it a bit more.
you really need to say whether it's solubility in water or
Or
in
lipid.
lipid.
Okay, gotcha. I think in general, proteins have very low water solubility. So if you want to inject therapeutic amounts of protein into your body, you actually need to inject a lot of solvent. You need to IV drip and everything. So if you want to increase the water solubility of proteins, polyethylene
That's
glycol
not going to happen.
Yeah. So yeah, you gotta. There must be new research into how you can incorporate these chemical groups, some sort of chemical groups, into helping the solubility problem, both in water and in.
Well, but what you're doing is quite clever. I mean, to bind it with the T cell and B cell.
Yeah. Well, not what I'm doing. I mean, that's what the research is doing right now. Yeah.
So which way do you confirm the right chemical modification in your protein?
So what sort of analytical methods?
How to confirm?
Yes.
So
this is definitely one of the key areas the industry is focusing on right now. As you can see, this is the analytical method is one of the key driving forces to streamline for the industry. Currently, a lot of people use mass spectrometry. So they would do a mass spectrometry on a protein, and if they can see a difference in mass that equates to the chemical modification, then that is correct. In parallel to that, an orthogonal test would be to do a trypsin digest. So what they do is they digest a protein, and then they fly each segment through the mass spectrometry. And if they know that segment contains that site, that contains that modification, then they can confirm it has been modified. And lastly, they can do crystallography if they're lucky to get protein crystals. But yeah, there is currently a lack of analytical methods to confirm site-specified modifications on proteins.
Well, there's also NMR as well.
Yeah, there's
NMR actually detect the environment of the hydrogen atom.
Mhm.
So that's a physical method.
That is but most of them we do solution NMR of proteins. So that can be done, but that cannot be done alone. So it must you must do the mass spec process as well in tandem.
So most of this kind of protein is small enough for MR analysis, right?
They can be done on NMR.
Yeah.
I'm not sure about but probably there must be some sort of technique to use NMR for antibodies as well.
Can I ask another question? Is that easy or possible to develop? Of course, it's possible to develop a very robust method to chemically modify certain residue group in the protein.
So the methods that I've talked about in this talk are three of the most general ways to do it right now. I think to be absolutely certain you get site-specific incorporation, you have to use a natural amino acid incorporation approach. So.
Or their mixture, you have to purify.
Yes, you don't want to be. I mean, if you look at this, you really don't want to be purifying a complex that contains 780 different possible regioisomers, because the two drugs. So if a drug to antibody ratio is 2, both two drugs can be both here, or could be both here, or can be here and here, here and here, it can be anywhere. So the regioisomer problem is very challenging. So to be absolutely certain you get site-specific incorporation like these or this, you would have to use a natural amino acid incorporation, because this would allow you to put in unique functional groups that are not present anywhere else in the antibody. And then it would have a unique reactivity towards your drug conjugate.
Okay, how do you introduce the
a natural amino acid?
acid?
Yeah, that would take me another half an hour to.
by some very special, advanced.
No, there is actually a lot of research being done in the field already. What they actually do is they take in mutant protein transfer. So they make use of the translational process of bacteria or animals, and they put in amber codon suppression transferase in order to incorporate such a unique amino acid inside. I can give you a few more references later, but probably beyond the.
Is that technique is mature enough to let people do this kind of work?
Yes, so this technique has actually been done by a lot of biotech companies already, startup biotech companies such as Allozyme and Brix. And the National Cancer Institute in America also uses this technique to investigate antibody drug conjugates. But I have to stress that there are currently only 3 antibody drug conjugates out in the market, and they are all done via unselective conjugation. So if people can discover a way or to mass produce this sort of technique, then I think it would be a huge potential within the industry. But it can be done. It can be done. I've done this before. So personally, in a lab, I've done this a natural amino acid. But I mean, I do it in a very small scale. Nothing that is good enough for, say, the pharmaceutical industry. But if I can do it, I'm sure some people will do it as well. Yes.
But what sort of scales you can go up to for the productions?
For the production of, so currently, one of the limiting factors of industry is getting enough proteins to meet the patient's demand. The most that I can see, they are making kilograms, but using like months and months to make kilograms. But because of its high specificity and efficacy, you don't actually need as much protein drugs as you would compared to, say, a generic drug. You don't need 500 mg paracetamol or something to get painkiller effect. You can use 1 mg or 2 mg of protein. But obviously, different proteins react at different, ways. So I'm just generalizing here. Say for sure.
Well, I would be generalizing as well. You could probably use a visual virus to insert that into a bacteria, to make a bacteria, well, in the bacteria's DNA to make it, well, I mean, that's basically how.
Recombinant method.
Yeah. Well, the recombinant method, yes.
That's
So that's the first
do a
class
lot
of.
drugs in mass scale.
Yeah. So if you look back here, most of the first functional class, the
Yeah.
recombinant proteins, these are done by bacteria.
Yeah.
So they
Yeah,
can
that's
be
basically
big.
how
You
it's
can
done.
make grams or probably even kilograms if you have large enough processed plants and stuff. But if you're talking about antibodies now, that's a bit more challenging. You probably don't get kilograms of antibody being manufactured on a week-to-week basis. It's a bit more challenging.
How stable are these antibodies?
So stability is another issue. So I would say they're not that stable. So they need to always be kept at a very cold temperature. They must be administered as soon as they've been defrosted. So there will always be research into attaching different chemical groups onto your antibody in order to increase its stability and to make it a lot longer lasting.
Well, they're not very stable just because, I mean, by their own nature, they are
They
biologically active molecules.
Yeah.
So they are.
and I mean,
well, they
they're
react to
huge.
bacteria, and also
They're
they-
huge. A little bit of the Herceptin, if it breaks down, then the biopharmaceutical property would be gone. So it's much more challenging than traditional small molecule drugs.
Well, I think there's another way about chemistry to do the conjugation is to express protein with a tag.
Yeah.
So how do you compare with this chemical method with that?
So the tag would be something like this, is it? So if you have if you have introduced this tag LLQGA into your antibody, then this transglutaminase would recognize this tag and then enzymatically conjugate this amine onto the glutamine group. Is that the sort of tag you're talking about?
I mean, the tag is kind of amino acid sequence that you express the protein with.
So this amino acid sequence, LLQ.
Yeah, so you mean this is part of...
This is another way of making it using enzymatic conjugation.
Yeah, so I mean, how do you compare this two methods? Like,
How do I compare? So I wouldn't say that there is one method which trumps every other method. But every method can complement each other. Some antibodies would be difficult to find a place where you can incorporate this peptide tag into. So if you use the traditional thiomab, where you don't actually need to incorporate any sort of peptide tags, that might be slightly advantageous. But if your drug molecule can only be made with amine molecule or that sort of molecule, then having the peptide tag might be beneficial as well.
I don't know how pure the products could be using the chemical method. Maybe,
Yeah. So
maybe with the tag, you can have a very purified. Because you know there's only one thing on it. But with the chemical, you showing that you can
be
be
done.
very sure you have like two or few tags, but maybe there's still a lot of molecules. Do you have many more?
Yeah, I think what people in industry use to purify proteins after a chemical reaction now is to use the standard membrane filtration, sub-size filtration, because these molecules or these sort of conditions, they are actually much, much smaller than your protein here. So if you just centrifuge your product through a membrane that size excludes, then your outcome would still be relatively pure. Obviously, there's other purification methods to purify your reaction afterwards as well. But you're right, I think using the peptide tag approach would eliminate that problem, but it's an extra protein engineering step. So, but I think, yeah, all these three methods are heavily used when doing research for antibody drugs.